All right. Is everyone settled in yet? Taruh di. Okay, I guess we're all settled now. All good? Okay. So good day, everyone. Thank you for coming here today. I'm Alicia from the International Office. Hope you're all doing well. So welcome to our Powertalk session for today, which will be on the topic of insulin and diabetes, presented by Dr. Graham R. Christie, Senior Lecturer and University Academic Lead for International Activity in the ASEAN region from the University of Dundee. Thank you so much for coming today, Dr. Christie. And also for today's Power Talk, we have Dr. Jessica Ginting, our very own lecturer here at the Faculty of Biomedicine, as our moderator. Thank you, Ms. Jessica, for being here today. Let's give them all a warm welcome. Okay, now there's going to be an attendance and evaluation form that you will need to submit for today. So do please scan the barcode for that attendance, which will be provided at the end of the session. And okay, without further ado, I give you Dr. Graham as our speaker and Ms. Jessica as our moderator for today. The floor is yours, Dr. Graham. Ms. Christie, Ms. Jessica.
Okay, thank you very much, Alicia, and thank you, Dr. Jessica. It's great to see you all, and it's a privilege to be back here at i3L. I think I first visited i3L back in just before the pandemic. It was in February 2019. So it's great to be back. As Alicia mentioned, I am a senior lecturer based in the School of Life Sciences at the University of Dundee. And Dundee is a small city located on the east coast of Scotland. Now, myself, I am Scottish, so hopefully you can understand my accent. Yeah, I'm trying to speak slowly so that I'm not confusing you too much. Okay. So today, as introduced, I'm going to be talking about insulin and diabetes. And I understand from Dr. Jessica that some of you are just learning or starting to learn about the endocrine system. So it's good timing for my visit here this afternoon. So I'm just providing an overview of what I'm going to present today. These are some of the key themes or sort of take-home messages that I'm aiming to cover. So I'm going to introduce, first of all, the topic by saying why is blood glucose control important? How is it regulated? What cellular mechanisms are involved in the secretion and action of insulin? The physiology to discuss diabetes mellitus and how that can be prevented and also treated. And then I'm going to present some research that was conducted just over a year ago that links COVID infection and incidence of diabetes. And then I'll wrap up with some relevant research undertaken at my home institution in Dundee. And then finally, we have the survey that you have to complete. And I'm happy to answer any questions that you may have. So that's what I intend to cover. A reasonable introduction, as I'm sure you're well aware, the cells of the body typically use three types of fuel molecules in order to generate energy in the form of ATP. And these three sources are fats, proteins, and carbohydrates. As a personal experience, nutrient availability typically fluctuates on a daily basis. So we find usually what we eat instantly, we tend to being digested and the nutrients are absorbed. We're going to have high levels of those nutrients within our bloodstream. And then over time, those nutrient levels will start to dip until we have our next meal, and that cycle continues. So we have fluctuation between two states, known as the absorptive state. So that's after a meal, as that meal is being digested and absorbed. That's when the levels of those nutrients are going to be high within the blood. Then in the post-absorptive state, that's when we have that dip in the concentration in the blood of those key nutrients. So the very fluctuation in nutrient levels, and those levels are also affected by things such as illness, infection, or physiological stresses, including exercise. So the human body is very much a dynamic environment. That really is the underlying principle of physiology. And each cell has the ability to switch the substrate used for energy production depending upon availability. I'm sure we've learned already about some of these metabolic reactions that are listed in the table at the foot of this slide. So we have things such as glycogenesis, that's the synthesis of glycogen, that will occur during that absorptive period when glucose is taken into muscle cells and then converted into glycogen. Glycogenolysis, that's when the glycogen is broken down to glucose, and that glucose can be released back into the blood. And then similar metabolic and anabolic reactions for the other nutrient fuel sources, proteins and fats. So I'm going to focus primarily on glucose and blood glucose control. So glucose is the major fuel source for most cells in the body. And the rate at which cells take glucose up from the blood is dependent upon the blood glucose concentration. So the higher the blood glucose concentration, the greater the ability for cells to take up that glucose, and vice versa. So it's maintained within a fairly narrow concentration range, typically anywhere between 4 and around 8 millimoles per liter. So that's the normal typical physiological range. looking at before meal is ingested, so at the end of a post-absorptive period, higher level is about 8 millimoles per litre immediately after the meal during the absorptive period. Now, it's important that we maintain blood glucose concentration within that range because there are some specific cell types within the human body that can only use glucose as their primary fuel source. That includes red blood cells, but more importantly, cells of the central nervous system. So it's important that these nerve cells are receiving enough glucose in order to generate ATP. Clearly, if the blood glucose concentration drops for any significant period of time, we're starving those important cells of the central nervous system, and that clearly is going to have quite a significant impact on physiological function. And it's been shown that it can be fatal if blood glucose levels drop to less than 3 millimoles per litre, even just for a few minutes. So glucose is important. It needs to be maintained, or the concentration of glucose needs to be maintained within that fairly typical and narrow range. So this is where the hormones and the endocrine system play an important role. Now, over time, we've a number of mechanisms and several different hormones that will act to increase blood glucose concentration. So I've listed some on this slide here. Some hormones that act to increase blood glucose concentration include glucagon, adrenaline, cortisol, and growth hormone. However, lots of hormones that will increase blood glucose concentration. There is only one hormone that acts to decrease blood glucose concentration, and that hormone is insulin. Now, if you think about this in terms of an evolutionary perspective, it's perhaps only in the last 50 to 100 years that food has become plentiful. Yeah. Before then, people had to hunt or scavenge for food, so food was scarce. So our bodies naturally are adapted to conserve resources, including fuel sources, with excess of nutrients. So remember, there is only one hormone, and that is insulin, that acts to decrease blood glucose concentration. So if we're consuming lots of food, ingesting lots of calories, there's going to be lots of nutrients available within our blood. We need a hormone that is going to regulate that blood glucose concentration. So I'm going to focus on insulin. Can anyone tell me what chemical type of hormone insulin is? Does anyone want to volunteer an answer? So we can subdivide the hormones in the body according to their chemical nature. And there are probably three main classes of hormones. Can anyone tell me the class that insulin falls under? Yeah. It is an endocrine, it's a hormone, you're right, but what kind of chemical structure has insulin? It's a peptide hormone, excellent, yeah. So insulin is a peptide hormone. The two major chemical types are the steroid hormones, so things like the sex hormones, testosterone. Examples of hormones are the amines. These are derived from specific amino acids, and these include adrenaline and noradrenaline. So insulin is a peptide hormone. So the next question, where is it produced and secreted from? Yes, it's from the pancreas. Specific. So the islets of Langerhans, we have these clusters of endocrine cells within the pancreas, correct? But there's a specific cell type within those islets, within those clusters of cells in the pancreas. And I'm telling you, it's the beta cells. Excellent. Okay, so it's a peptide hormone secreted from beta cells within the pancreas. Now, its primary for secretion from those beta cells is an increase in blood glucose concentration. So during that absorptive period, so once we've digested a meal, that meal has been broken down into its constituent building blocks, its nutrient building blocks, and those have been absorbed into the blood. We have an increase in the blood concentration of those nutrients, including glucose. You can see on some later slides, I'll show the cellular mechanisms that links that increase in blood glucose concentration to the secretion of the hormone insulin. Then we'll tell me what the target tissues are for insulin. So insulin is a hormone that forms part of the endocrine system. That hormone is transported in the blood once it's been secreted from the pancreatic beta cells. That means, as for every hormone that's secreted, all cells in our body are exposed to all circulating hormones. It's only the cells that express the receptor for that particular hormone that will elicit a physiological response. So the target tissues are the cells in the tissues of the body that express that insulin receptor. So can anyone tell me which tissues or organs in the body insulin acts upon? Yeah. It also acts on the liver, so that's one major one. Yeah. It does, yeah, but there are different types of muscle cells, aren't there? So skeletal muscle cells, yeah, excellent. So those are two. There's maybe one and third major one. Yeah. Adipose tissue, fats, excellent, well done. So the target tissues for insulin, liver, skeletal muscle, and fat cells. Now, its physiological actions I'll come on to in a later slide. So back a step, we said that insulin is secreted from beta cells located within the pancreas. As I said, within the pancreas, we have clusters of endocrine cells. These are called these islets of Langerhans. And this figure here is taken from a Nature review, just compares the islet form of that of human. So the panel A, human, on the right-hand side. And then we've got a table that summarizes the cellular composition of both the rodent and human islets. We'll focus on the beta cell. That in the rodent, we have a higher proportion of pancreatic beta cells compared to humans. Well, this is important because, as you know, as a trainee scientist, a lot of experiments are carried out on rodent models, mice or rats. Now, it tells us that there is a difference between rodents and humans. So it's important to take these things into account when using such experimental models and inferring or transferring the results from those models to human subjects. Here we've got an image of a pancreatic beta cell. Some of the steps involved from glucose at high concentration in the blood to secretion of insulin. And sort of this picture here, the plasma membrane of the beta cell, we have a glucose transporter. It's a membrane transporter that's specific for glucose molecules. It's called GLUT2. It's a facilitated glucose transporter, so it relies on an existing concentration gradient across the cell membrane. With a high blood glucose concentration, lots of glucose outside the cell, That glucose is then subsequently transported into the beta cell via that group-2 membrane transport protein. I can show that glucose is then metabolized in the process of glycolysis to yield ATP. So when glucose is transported into the cell burns that glucose and generates ATP. It causes closure of another membrane protein. This time it's a channel. And it's a channel that's specific for potassium ions. Now, if we close off that channel, it stops potassium ions from entering the cell via that route. That causes a change in the membrane potential. We have a slight and subtle change in membrane potential. That change in membrane potential, in turn, opens the voltage-gated calcium channel, and that's shown down here. So the change in membrane potential opens a calcium channel, and calcium ions enter the cell. And it's that calcium ion entry that in turn acts as the trigger for release and secretion of insulin from the pancreatic beta cell. So glucose comes in, glucose metabolized to ATP. ATP closes that ATP sensitive potassium channel. That causes a change in membrane potential. Calcium enters, and that calcium triggers secretion of insulin from the beta cell. Look at the right-hand side of this form, insulin is then transported around the membrane, and it reacts on the target cells, i.e., the cells that are expressing the insulin receptor. And what receptor the insulin receptor is. You covered this in your physiology or biochemistry. It is a tyrosine kinase receptor. So when insulin binds to this receptor protein, it phosphorolates itself. Okay, it adds phosphate groups to specific residues on that receptor structure. And that phosphorylation in turn acts as the trigger for a number of different cell responses. It's shown in this figure here, using a skeletal muscle cell as an example. What happens is we get translocation of a glucose membrane transport protein related to group 2. This one's called group 4. If the insulin binds to the muscle cell, we get insertion of group 4 glucose transporters in the plasma membrane. But if we have transporters for glucose within the plasma membrane, that means we've increased the capacity for that cell to take up glucose from its external environment. So if you think about that in a stepwise process, insulin binds to the skeletal muscle cell. The response to that is we have more glucose transporters. That means these cells are able to take up more glucose. So what's going to happen to the glucose concentration? It's going to be So starting off eventually with an increase in blood glucose concentration after ingestion and digestion of our meal, that triggered the release of insulin from the pancreatic beta cells, which are acting on our skeletal muscle cells to increase glucose uptake. Now, if you think about your body composition, most of your body is made up of lean tissue and also fat tissue. And if we're stimulating glucose uptake into your muscles, that's going to have a significant impact on blood glucose concentration. We're going to lower blood glucose concentration. One interesting fact about that group 4 transporter is that expression in the plasma membrane is increased when we exercise. Now, as a physiologist, I'm interested in exercise physiology. So when we exercise, we also increase glucose uptake into our skeletal muscle cells. And this is through increased GLUT4 expression, but via a different pathway from that stimulated by insulin. And then there are some other effects that insulin has on our skeletal muscle cells, with the regulation of human metabolism, which I'll come on to, and we can also determine gene expression and cell growth. So there are a number of effects on skeletal muscle cells in response to insulin. Let's summarize those key points. The effect that insulin has on carbohydrates is that it promotes glucose transport into skeletal muscle cells. It also does the same in our fat cells. So collectively, increased glucose uptake into those tissues is going to decrease blood glucose concentration. The glucose that's taken up into skeletal muscle cells is then converted into glycogen, and insulin stimulates the enzymes responsible for that metabolic pathway. And as we're synthesizing glycogen, we don't want to be breaking it down at the same time. That would be counterproductive. So what insulin also does is it inhibits the breakdown of glycogen. So insulin stimulates glycogen synthesis and turns off the breakdown of glycogen. Then some other effects on fatty acids. It stimulates fatty acid uptake into fat cells, and it promotes the conversion of those fatty acids into triglycerides. So again, we're taking the basic building blocks and storing them within the fat cells as triglycerides. At the same time, we want to be inhibiting the breakdown of those triglycerides. The acids, the building blocks of proteins. Again, insulin is going to stimulate amino acid uptake into skeletal muscle cells, and it will also use those amino acids to build new proteins through increased protein synthesis and inhibition of protein degradation. So we're taking the building blocks, we're storing them inside cells, and we're converting them into storage molecules. That's essentially what's happening in response. Action of insulin. Consider diabetes. Diabetes, I'm sure you know, is a chronic condition. Because when either the pancreas doesn't produce enough insulin, or it is producing insulin as normal, but for some reason that insulin is not acting as it should on those target tissues. So individuals that suffer from uncontrolled diabetes will have raised or elevated blood glucose concentrations because the insulin isn't being produced or that insulin isn't acting as it should on those target cells. But over time, that increased or raised blood glucose concentration can cause quite serious and significant damage to many of the body's tissues, particularly nerves and very small blood vessels. There are three types of diabetes. Type 1 is the one where individuals are unable to produce insulin. So these are usually the diabetics that require insulin injections to regulate their blood glucose concentration. And diabetes occurs when there's a breakdown of the beta cells within the islets of Langerhans. And sometimes that's due to an autoimmune response. So for some unknown reason, that individual is producing antibodies that attack and break down their own pancreatic beta cells. And clearly, if you don't have any functioning beta cells, you're not going to be able to produce insulin as normal, and your blood glucose concentration is going to remain high. most common type of diabetes is the type 2 diabetes mellitus. So this accounts for more than 90% of all diabetics. And type 2 diabetes, these individuals are producing insulin. For some reason, that insulin is not acting on those target cells. Common causes of type 2 diabetes are linked to excessive body weight and physical activity. And type 2 diabetes progresses over a long period of time. It's not something that just happens overnight. It develops over a longer time scale. That's something that I'll come back to. Type of diabetes that I'm not going to go into any great detail is gestational diabetes. So that leads to increased risk of complications during pregnancy and during birth. It's been shown that mothers that develop gestational diabetes, their child then has an increased risk of developing type 2 diabetes. So there is a link between gestational and development of type 2 diabetes in their offspring. So how do we diagnose diabetes? It's relatively straightforward. We simply have to test blood glucose concentrations. So there are three sort of main tests. There's a random test, a fasting test, and a 2-hour postprandial test. The fasting one is probably more common. Your doctor will ask you to fast overnight. You'll come in so you'll take a blood test at the start to determine your baseline blood glucose level at fasting state. And then give you a little dose of glucose, and then at regular time points thereafter, you'll take further blood glucose tests to determine your blood glucose concentration. Depending on the concentration and the test, you can then get into one of these three major categories. You either have normal blood glucose control, you're diabetic, so you're in the route to developing type 2 diabetes, or indeed you are a diabetic. So I mentioned the health impact of hyperglycemia. So this can damage the heart to the blood vessels, the vessels to the eyes, but also kidneys and nerves can also be damaged. Some stats there listed there. As a result of this, adults with diabetes have a 2 to 3-fold increased risk of heart attack. There's increased risk of neuropathy, that's nerve damage in the feet and the extremities that can lead to foot ulcers, infection, and possibly even limb amputation. Damage to the small blood vessels at the back of the eye as a result of diabetes is an important and common cause of blindness. And diabetes is among the leading causes of cancer failure, kidney failure. Collectively, I think we can appreciate that diabetes causes a marked increase in both morbidity and mortality. So to back up this, there's some data from the International Diabetes Federation that shows that 1 in 10 of the global adult population is living with diabetes. This is something that's on the increase. So by 2030, it's expected to be 1 in 9 of the adult liver population, and by 2045, 1 in 8. There are some other statistics listed there, including the just and healthcare implications for the treatment and management of individuals with diabetes. Some stats that I've pulled out about Indonesia. Calculations, the adult population in Indonesia is about 180 million. 80% global figures from 2021 of the adult population in Indonesia have diabetes. What's quite scary is there is at least another 8% of the adult population that are currently undiagnosed. So 11% to 8%, a significant proportion of the adult population either have diabetes or undiagnosed diabetes. In terms of treatment, I mentioned earlier that for type 1 diabetes, we can replace the insulin that isn't being produced naturally by the body by insulin injections. Injections regulate blood glucose. Some drugs that can be used to help with that. Some may also require insulin injections. It's also important that diabetics monitor and regulate their blood pressure, going to try and reduce the incidence of cardiovascular disease. It's important to look after their feet because remember, with the damage to the blood vessels in the extremities, that increased risk of developing foot ulcers. It's important that they have regular checks on their eyesight, just to make sure that they're not developing that retinopathy and subsequent blindness. And we need to make sure that the kidney function is also monitored. So these are just some of the interventions involved. In terms of treatment, key is looking after your diet and engaging in physical activity. I mentioned previously that insulin causes an increase in expression of those GLUT4 membrane transport proteins. And I said that exercise also does the same. So it's been shown that if an individual, let's say, has type 2 diabetes and undertakes some form of regular exercise, that can help contribute towards their blood glucose control and removes the over-reliance on medication. For all conditions, prevention is better than cure. So it's important that individuals try to achieve and maintain a healthy body weight, try to be physically active, eat a healthy diet, and avoid tobacco use. So these are all things that we all know about, but in practice, it's sometimes not particularly easy. So that covers hopefully blood glucose control, insulin, diabetes mellitus. Next slide. This is the study that was conducted, I think, in March last year, where we did a data analysis looking at 180,000 people who had been infected with COVID and compared outcomes against 4 million control subjects. And we found that those that had been infected with COVID, 1 in 100 developed type 2 diabetes. So there's 1 case of diabetes for every 100 cases of COVID infection. Back to the earlier stats that I showed, that already 1 in 10 of the global population have diabetes. 95% of those have type 2 diabetes. But if you think about the COVID pandemic and subsequently, because COVID hasn't gone away by any means, those that have been infected with COVID, 1 in 100 of those have that increased risk of developing type 2 diabetes. So what we have is a major global issue with individuals suffering from diabetes. We've had a COVID pandemic, and it seems that there is a direct link between COVID infection and developing type 2 diabetes. So again, this is going to have major health, societal, and economic implications. So medics, nurses, it's important that they are checking on individuals that have suffered from COVID, just to make sure that they're not developing type 2 diabetes. Clearly, there's lots of research going on looking at diabetes, its causes. It is no exception. Here we have just a couple of studies published in the last year or two looking at aspects of diabetes. So this one here on the right-hand side, it's been shown that South Asians are up to 3 times more likely than Europeans to develop young onset type 2 diabetes. So lots of work going on in my institution looking at the links between genetics and diabetes. So to summarize my presentation, we've looked at the endocrine control of human metabolism, how that's important, how insulin is the only hormone that acts to lower blood glucose. That's important because after the meal, we're going to have high blood glucose concentrations. It's also important because it says if we have high blood glucose concentrations over a prolonged period of time, that individual is therefore diabetic, and there's all sorts of consequences as a result. Just lastly, then, at the link between food infection and also diabetes, and how this links to relevant research at my home institution. So I think that pretty much concludes my talk this afternoon. Here is the QR code to your evaluation survey. This afternoon, I'm going to ties in with your studies and for your attention and attendance. It's been great to see you all, and I'm happy to answer any questions or queries. I'll try and answer those as best I can. Okay, so thank you.
Okay, thank you, Dr. Graham. Now, well, actually, I will also do some Googling on the Indonesian prevalence number for diabetes. Actually, International Diabetic Federation ranked Indonesia as sixth highest, and our population actually stands 230 million. Yeah, so 10.9% is diagnosed with diabetes. Yes, according to National Health Survey. So it's basically increasing every year. So that's except the undiagnosed one. Okay, so that's quite
very significant, isn't it? Yeah.
Okay, so now I will invite questions from the students, especially if you have any questions. This is a very, rare opportunity to ask the expert. Okay, please, Matthew, right? Okay, please, Matthew.
So, I was wondering how that get passed on to the generation.
Question, yeah. So clearly there must be some sort of genetic link there. I think type 2 diabetes is very much a good example of a multifactorial condition. It's not going to be due to just one particular underlying cause. It's going to be things multifaceted, all working at different points in time. And I think as a result, that makes it very difficult to pinpoint exactly what the underlying cause is, because there's not one. Clearly, there are going to be some genetic elements there, but also there's going to be lifestyle issues at play as well. So a combination of genetics and environmental factors will make it very complicated. I think key is, if you're aware of the family history of diabetes, as for any condition, is to make sure that you are trying to manage your diet, trying to undertake exercise regularly, just generally looking after yourself. Also making sure that you are being checked at regular intervals, just to make sure. Type 2 diabetes, if you're in that diabetes stage that I mentioned on the slide when we looked at how do we detect diabetes, it is possible to reverse that. And again, there's been lots of studies showing that in individuals that undertake fasting diets, that has been shown to have a significant impact on reversing the development of type 2 diabetes. So Again, I think it's catching it early enough and then intervening at that point so that you've got an opportunity to revert back. Thank you.
Okay, so clear and clear. Okay, is there any more questions, please? Yes, please to mention your name. Yes.
Yeah, so yeah, I think there are groups of people now that can help to a certain extent, but they're not by any means a magic bullet. They're not going to type 2 diabetic that has difficulty controlling their blood glucose levels. It's not going to fix that problem completely. They're still going to have some issues with that. So for me, I think that's the biggest problem. I think often when patients are prescribed a drug, we think that that's going to be a complete cure. But that's not really the case here. And I think for a lot of clinicians, there's a lot of trial and error with individual patients to find, first of all, the treatment that works best for that individual. Because again, as we know, drugs work the same in each individual. So it's trying to personalize that treatment, but also to get the dosage right for that individual. I think personally, key is trying to manage your diet and have regular physical activity. I think physical activity is something that many of us struggle to fit into our daily routine. And I know it's not easy, but it does have such a wide range of health benefits, not just for glucose control. I think there was a study just published this week that linked physical activity was better as a cure for those with depression than some pharmaceutical agents. So yeah, thank you.
Okay, you first, you next. So, yep.
If you look at the practice by 1.5,
Okay. I think that would be unlikely because type 1 diabetics are normal when they don't have any insulin. They're unable to produce or secrete insulin, and that's due to a defect in those beta cells. Type 2 diabetes is normal when the individual is producing insulin, but that insulin isn't working as it should within the body. So yeah. Type 1 diabetes tends to develop at a younger age. It tends to develop in younger adults, teenagers. And again, it's usually triggered by that autoimmune response. There are some links between certain infections. For example, I think glandular fever is one. There's been some studies to suggest that if you're infected with glandular fever, you have a bigger chance of developing type 1 diabetes. Now, not in all cases, but there's an increased risk there, I think. Yeah. Diabetes is more slower onset and again linked to being overweight and physically inactive.
That answer your question? Okay, so I briefed this. Okay, so many questions. Sorry, miss, how many minutes? How many? 10 minutes. Okay,
10 minutes.
okay. First, you in the back. Okay. Please continue. Thank you. Okay.
Excellent question. Good question. Okay. So you can much more physiology in the program. Not yet. Okay. So what happens in rooms with high blood glucose concentrations is that the kidneys filter the blood. And through that process of filtration, there is then a process of reabsorption. So within the kidneys, nutrients that are filtered are normally reabsorbed back into the blood. So normally, we don't excrete any glucose in the urine. In diabetics that have high blood glucose concentrations, the kidneys are filtering that excess glucose and unable to reabsorb all that filtered glucose. So the kidneys, in terms of the absorption, are working at full capacity, but unable to reabsorb all the glucose back into the blood. And as a result, some urine ends up being excreted in the urine. So that is an easy clinical test, is to have one of these dipsticks sensitive to glucose and test the urine sample. And if that comes up positive, then there's a good likelihood the individual is diabetic. So it's to do with the capacity of the kidneys to reabsorb glucose. If your blood glucose control is normal, your kidneys are able to filter and reabsorb all that filtered glucose. If you have high blood glucose concentration, your kidneys are unable to reabsorb all the filtered glucose, so some ends up being excreted. Good question. Yeah.
Thank you, Doctor. Is that answering your question? We will have that experiment after the term for HSF lab.
Ah, excellent. Yeah.
Okay, and I think this. Okay, I haven't reached this part. Okay, okay. Who's first? Who raised? Okay, you. Okay, okay. You first. Please mention your name.
So artificial sweeteners for those with diabetes. Yeah. It's a good question. To be honest, it's not something I know the answer to. So I'm not sure. Does anyone else? Do you have a view? Is it something that you use yourself, or do you use artificial sweeteners?
Lot of papers also it will be influenced, but I think I also look one of the
Yeah. So conflicting evidence in the literature. It's often the case with nutrients and studies looking at nutrients. It's very difficult to conduct studies using human subjects as it is, but getting a group of subjects to follow the dietary regime and compare that against another group working on something different, it's very difficult to do that properly and compare the results. So yeah. Some studies suggest that certain sweeteners can change the regulation of nutrient transporter expression within your GI tract, within your guts. There's one particular sweetener, I can't remember the name, in some of the fizzy drinks, the diet fizzy drinks, that has been shown to affect nutrient uptake.
But I think in diabetes, actually, the most important is to reduce the sugar consumption, not to replace the sugar, but to reduce sugar consumption. I think that's the best that we can do now, because researchers said, yeah, few of the artificial sugar, they give negative effects to our body, so it's better to reduce the sugar consumptions instead of replacing it. Okay, I think we still have 3 more minutes, maybe 2 more questions. Three more questions? Oh, okay, 2 more questions. Okay, this is going to stop. Okay, no. Okay.
My name is Michelle. I'm curious about, actually, I read some papers about gut dysbiosis before that leads to chronic inflammation, hence the resistance towards insulin.
Yeah.
Just now you mentioned about focusing on diet. Is it possible if we eat prebiotics or probiotics, or maybe even fecal transplant? Because I don't think the research on that is very well developed right now. What are your thoughts on that? Thank you.
Okay, I didn't quite catch the full question, but it was about inflammation and linked to diabetes, is that right? And probiotics?
Yes, that microbiota.
Gut microbiotics, yeah. I think going back to that study linking COVID infection and development of type 2 diabetes, where there was 1 in 100 I think that must be linked somehow to inflammation. So going back to the earlier question about type 2 diabetes, again, there are links there. And we know COVID infection leads to, that increase in inflammation. So there's definitely some sort of link there. As for probiotics, I don't think it's going to do any harm. But again, it comes back to the previous question about studies looking at nutrients and nutrient intervention or dietary intervention in this case. It's very difficult to conduct those studies properly in human subjects because there's just so much variation and it's difficult to get human subjects to adhere to a particular regime for a long enough period of time. So yeah, but I think it's probably not going to do any harm. Yeah.
Okay, does that answer your question? Okay, so I think we will leave the last two questions for online questions. You have questions? Okay, I'll save the last questions for me with that, especially for me. Okay, so we have two questions online. The first question is from Nicholas. He asked, has there been any innovation in exogenous insulin prescription? For example, adjuvant prescribed along with insulin or structure to increase the insulin activity?
Wow.
Oh, sorry, efficacy, efficacy, not activity.
Yeah, excellent question. Again, it's not something that I know a great deal about, I'm afraid, and so I can't comment specifically. About GLP-1 developments that we use to control weight loss. These are drugs that mimic a GI hormone called GLP-1. GLP-1 is an incretin which acts to increase insulin secretion from beta cells. So it was originally derived and used to treat individuals with type 2 diabetes. It was then discovered that those individuals were also losing weight. And that is because GLP-1 has an impact on the central nervous system and regulates diet, regulates hunger pathway. It's been shown that individuals that take these drugs can lose up to 15% of their body weight over 12 months. So these types of drugs are being marketed specifically by the pharmaceutical industry to target weight loss rather than type 2 diabetes. Yeah, there's quite a few Hollywood stars, I think, claim to be taking these sort of weight loss drugs. Yeah.
So, but it's very expensive.
Very expensive, but drug companies are making some money out of it. Yeah.
That's bad. I might need it. Okay, so next question is from Kanye here. He asked how cases of insulinemia or pancreatic beta cell tumor will take effect in patients with diabetes, or will the other effects will present earlier, such as early onset diabetes, like solutions. So how cases of insulinemia will take effect in patients with diabetes, sir? Insulinemia.
Just trying to dissect that question. There's lots of components to that.
How cases or the other effects will present earlier, such as earlier onset of diabetes-like conditions. How cases of insulinemia will take effect in patients with, or maybe if there's a tumor in the, for example, pancreas, will have an effect in patients with diabetes. Maybe that's the question.
I guess it depends on the tumor. There's lots of different examples of tumor affecting particular endocrine glands within the endocrine tissues that result in hypersecretion of particular hormones. I guess it depends on the type of tumor where it is. Yeah, it's going to be,
Okay, I think that's answered the questions. Okay, now I will give the last question to Ms. Swedet here, one of our faculty, sir, in biomedicine. Okay.
I hope this one isn't too hard. That's really challenging.
Yeah, thank you, Dr. Brown, for your presentation. I came here to the session because I'm personally interested. I suffered from gestational diabetes, and with diabetes after I gave birth to my son. And I'm still struggling because I have now I'm in a better state, pre-diabetic, but still it's an interesting topic for me. So I read a lot of research. So my question is, since type 2 diabetes is not inherited, or like some part of it is inherited, so I'm wondering, because it's not clear in research articles, whether the implication, what I know about type 2 diabetes, is that sometimes the receptor is defective, insulin receptor, or one of the proteins in the cell signaling of insulin. So is that possible that the genetic component of type 2 diabetes is related to a mutation in the receptor itself, or like, for example, to IRS, insulin receptor substrate protein?
I think a good question. I think there are so many components involved, going from insulin binding to that receptor to any of the cell responses elicited. There's probably hundreds of thousands of individual proteins involved in that cascade. Once it introduces changes in a few of those to disrupt the normal cell balance, which is, if it's amplified, and when I said that the target tissues are fat and skeletal muscle cell, it's amplified around a large proportion of your body composition. So that is probably going to have quite a significant impact on your whole body physiology. It's not like we're just acting a small number of cells in a small tissue or organ system happening across lots of cells collectively, resulting in a more physiological impact.
Is that possible that we inherit it? Because usually they are telling family history. I'm not fat. Sure, it's like they are saying family history. I'm not overweight, sorry. So I'm wondering where this is coming from. Like, must be, is it possible to inherit a specific gene mutation? In this case,
I think it's entirely possible.
type 2 diabetes.
Yeah.
But when I talk to doctors, they're saying it's complicated to explain.
I think, well, there's some potential points of genetic mutation.
Oh,
Yeah.
okay.
It's one of these things, I think that an answer, it's like type 2 diabetes. I think it's more about prevention. Finding the best treatment. I think
Okay.
that's going to be the case.
Thank you.
Okay, I think we are running out of time. Because I'm going to give it back.
Okay. Thank you so much, Dr. Graham Christie, for your insightful lecture today on insulin and diabetes, and Dr. Jessica for being our moderator for the Powertalk. I'm really happy to see the enthusiasm today from everyone. Thank you for your participation. And as presented by Dr. Graham, these things are being studied also at the University of Dundee. Yeah? And with that regard, I'm pleased to present to you our next session, which is the info session on studying in Dundee, which Dr. Graham will also present to you on the double degree program and the postgraduate studies.
Yes,
Sure. So stick around while we get set up for the info session. And in the meanwhile, please don't forget to fill out the evaluation on the evaluation and attendance barcode. Oh, okay. And in the meanwhile, itu jadi suruh pada ke bawah semua. Okay. Anyway, let's go to the front for pictures for now, or for documentation. Foto-foto dulu sembari dianya juga itu. Info session kalau bisa lebih baik, tapi kalau lagi nggak bisa nggak apa-apa juga. Okay, let's go to the front, everyone. For pictures? No. Okay. All right, everyone, if you could go to the front for pictures so that we can document your presence today. Okay. All right. Yeah. Okay, go ahead, go to the front. Mas Adit bisa tolong di itu enggak di apa tuh di
Let's go. Let's go down the middle here. Okay, make it quick. Okay, yuk semua turun yuk, satu yuk. Nick. All right, you got some of you, this over there. Can you move here, please? Fill in the gap. Just like make 2 or 3 lines, it's fine. Okay. Okay. One. It's bit up front. Yeah, so you can see it clearly. There you go. All right. From the shots, can you move here? Yeah, all of you guys. Just move over here. Yeah, move over here. Okay. Three, two, one. Another one. Three. Right. 3, 2, 1. Right. One more. 3. Right. 3, 2, 1. There we go.
All right, everyone, please be on standby while we prepare for the next info session for postgraduate studies and double degree at Dundee. Thank you.
Okay. First, we will finish you can still join for the info sessions where we will control and postgraduate studies. Okay, so stay here. If you want to that, stay.
Okay, together.
Right, thank you, guys. Okay, thank you.